let's go ahead and get started. A couple of quick announcements. Again, the first one has to do with our seminar this week. Biology doesn't have one, but chemistry does, which is then after the midterm. So you'll all be relaxed, or your brains will be mush, as the case may be. Should be a really interesting talk. Pam Cassidy from OHSU Dermatology is going to be talking about oxidative stress and melanoma, which actually ties in really nicely to some of the DNA repair stuff we'll be talking about right at the beginning today, again, getting back to the last one. The other one, and this is something that came up in my Twitter feed just last night, having to do with start codons, and I think someone asked me a start codon question that I answered on D2L, sort of which start codons are actually used. This is the case for bacteria, where AUG, GUG, and UUG are often used, which is, you can sort of tell by how brightly each of these things are, just looking at expression of a particular protein. But it turns out that if you look very closely, E. coli at least can use a whole bunch of other start codons. So it's by no stretch of the imagination just AUG, GUG, and UGG, whole bunch of other ones as well. And this was literally just published. Now, how relevant this is to other systems is not completely clear. But it is a sort of fascinating question, and it may well have to do with the fact that, again, we've got ribosome binding sites in bacterial systems, and because of that whole ribosome binding site, that's why you can use pretty much any codon as long as it's in the right place relative to the ribosome binding site. So that being said, again, this is a review session. All the slides are identical to ones that I've used before, with maybe one exception, because I found a mistake on one of my slides. So I will use that. But one of the things I realized at the end of last lecture is that we were missing something. And what might that something be? A clicker question, which is great review for that last one. So our clicker question, I haven't started it yet. So presence of which of the following on mRNA leads to nonsense-mediated decay? Cap binding complex, EF4E, EF4G, exon junction complexes, and EIF2. May I start now? 5, 4, 3, 2, 1. Go. Vote early, vote often, as in the best democratic way. Only the last vote counts. Ten, 4, 1. Okay, so question basically here has to really do with nonsense-mediated decay, and it's nice that I also get to review some of the other things here. Cap binding complex is where? Binding to the cap. Do you see it at all in translation? You shouldn't. Cap binding complex is really important for getting export to take place, but not about getting translation to take place. On the other hand, EIF4E does what? Binds to the cap for translation purposes. What about EIF4G? That's forms a bridge between the binding proteins at the cap, EIF4E, and the poly A tail. Makes your nice loop structure. EIF2 does what? Eukaryotic initiation factor 2. 6 of you thought it was right. No, so yeah, eukaryotic initiation factor 2. I always have to slow down with these things. Initiation factor 2 is also the same in bacteria. What's that for? Getting that initiator tRNA into the small subunit of the ribosome and associated with the start codon. And so presumably all those funky start codons that we just looked at, that they found that work in E. coli, all of those are presumably interacting with your initiation factor 2, because it's bacteria, and a initiator tRNA. So it can sit down on any of those particular anticodons. So of course, the only thing we have left, exon junction complexes. That is what happens when you have splicing. After splicing is taking place, you have these exon junction complexes. Normally, with translation, all of those get bumped off. If there are any that are left because you've stopped too soon, i.e. nonsense codon, you've got to stop codon too soon, then they're not removed from your messenger RNA, and that's the signal to give nonsense-mediated decay. Yeah?
So EIF2, on the slides, it says that plus EIF5B, I think, that is analogous, those together are analogous to IF2. Are you going to want us to know anything about EIF5B other than that it binds with?
So the question here, and I forget if it's one of the slides that I'm using as a review, it might be. So the question is, what about all those other eukaryotic translational initiation factors? So just to paraphrase your question. And the answer is, there are so many of them, I can't keep track of them all, so I can't really expect you to. However, the 1, 2, and 3, I think, are perfectly fair game. Which is also, if you may remember correctly, those are the ones which are red on the slide. So, okay, so let's rewind a lot and go back and talk about DNA damage. This is why I brought my little model with me again. This is, again, why DNA is a really crummy molecule to use for your genetic information, because it's incredibly susceptible to all kinds of chemical changes. Any kind of chemical change can change the base pairing interactions. Of course, you change the base pairing interactions, you've changed the genetic coding material, which is here. A couple of things that I wanted to specifically emphasize here, and some people asked me about this, I think, also on D2L. Most common DNA damage that happens, how big the arrows are. The biggest arrows are right here and over here. What's that? That's depurination. So it's hydrolysis of the glycosidic bond, which is between the base and the sugar, removing that base. By far the most common of any of the DNA damages that we're going to talk about. Second most common is deamination of cytosine, this arrow right here. We haven't talked about oxidative damage. Again, come to the seminar on Friday. I'm sure we'll hear a lot about different oxidative damages. Methylation also happens quite often in terms of DNA damage. This is, interestingly enough, the kind of damage that happens to your DNA if you consume large amounts of carcinogens, and best study is actually tobacco smoke will methylate various different parts of your DNA. So once you've had that DNA damage which has taken place, it needs to be repaired. Otherwise, we've got genetic changes and so on and so forth, which is good for evolution, but you don't want to have too many of them. So these are the two sort of main techniques that are used to repair now damaged DNA. What was the most common damaged form of DNA that I mentioned? It's this depurination. So it's actually right here. See? There's lacking a base in this particular structure. Here's depyrimidination, but depurination would give you the same kind of structure. So it's a double-stranded DNA, like we have here, that's missing one of the bases in here. So if it's missing one of the bases, then you have an AP endonuclease, which will cut, and it's just an endonuclease, so all it does is cut the phosphodiester backbone. Then you have to have a phosphodiesterase, which is going to cut out that sugar, which is still in the backbone. Now you've got a nice template and primer that DNA polymerases can elongate, which is great, but then there's still going to be a gap between your phosphate and your OH. Then you need a DNA ligase. So there are multiple enzymes that you have to have here. Now, of course, if you've got deaminated cytosine, or another one of the common base changes that's not depurination, now you have a glycosylase that basically gets you to this same step right here, which is, again, the most common kind of DNA damage that you have. So questions on base excision repair. So single base, that's what it's all about. Okay. If, however, you have something which is more problematic and also usually a lot rarer, then you have a change in the structure of your double helix. Change in the structure of your double helix, classic case is some kind of pyrimidine dimer. Then you have the nucleotide excision repair pathway, which is a lot less efficient, because what happens here is you have cleavage that takes place. So 2 endonuclease cuts, and then this piece of DNA is not just going to fall off by itself. You need a DNA helicase to take that whole strand off. Now, just like you had down here, only it's a much bigger gap, is a 3'OH, a template, and your DNA polymerase can replicate through that. Take care of it. This is a very general repair technique. You don't need a very specific glycosylase to take off that modified base, but any kinds of changes that can have happened to your DNA, at least as long as they haven't changed both strands, you can deal with this mechanism. And often there are these other big changes that can happen to your DNA molecule here. Again, pyrimidine dimers is just one example of that. Sometimes you'll have other things which have been bound to your DNA, big extra proteins, large phenyl structures, you name it. Any of these big things which change the structure of your DNA can be repaired through this kind of mechanism. Okay, more questions on this stuff? Again, I will continue to move through here. Double-stranded breaks. This is what happens, of course, when you have cuts on both sides of your DNA, and you can't have that backup copy, the template, which is already over on the other side. 2 basic ways that this gets dealt with. The quick and dirty way, which is non-homologous end joining, which has exonuclease activity that chews in the ends of your piece of DNA. Once those have been chewed in, then the ends have to be ligated back together. And one thing that I didn't go over in too much detail here, once you've done exonuclease cleavage, a lot like what happens in the case of base excision repair, you've got to make sure that you have an end which you can ligate back together. And so ligases have to have 3'OHs and 5' phosphates. And those then can be put back together. So it depends on this exonuclease activity. If you still have those phosphates, you're fine. If you don't, and most exonucleases cut them off, you're not in good shape. So this process is also what's being used, or at least originally, for CRISPR-Cas9 genome editing. I went and looked at the syllabus for after this midterm. I want to try and put in a lecture on CRISPR-Cas9. People interested in that? Yes? I will try and move some stuff around. Probably less transcriptional regulation, more CRISPR-Cas9, cool small RNA stuff. So I will definitely do that. But a much better way, better, i.e. not losing genetic material in this process of repairing double-stranded breaks, is homologous recombination. So homologous recombination also has this exonuclease activity, but because there's a homologous chromosome, you can use that information to repair where you had your double-stranded break in the first place. This is slightly dangerous because that homologous chromosome is not necessarily a duplicate chromosome. It's just a homologous chromosome. And we'll get back to that. That's this whole gene conversion thing that we have down here at the bottom. So how does homologous recombination work? You have a double-stranded break. This double-stranded break could be made by a endonuclease. So that's what happens in meiotic recombination. You have endonucleases that will make a cut. So that's how you get crossing over that happens in meiosis. But you can also have double-stranded breaks that form in lots of different ways, probably mostly in haploid organisms. It'll be a nick in your template strand, just like you have right here in our DNA. Oops, there you go. Phosphatase activity. And then when you replicate along, you'll end up with a double-stranded break. So that gets repaired, again, through this homologous recombination mechanism. First thing that happens is you have an exonuclease that chews in at the 5' end of your molecule. So it's a 5' to 3' exonuclease. This gives you a single-stranded piece. Together with Rec-A, Rad51, this then does strand invasion. So it goes and tries to find its homologous sequence. Yeah?
Can we think of Rec-A and Rad51 as kind of chaperoning this process? Or?
So the question is, can we think of Rec-A, Rad51 as kind of chaperoning the process? I wouldn't say chaperoning per se. You actually absolutely require those proteins in order to get strand invasion to take place. So when I think of a chaperone, it's helping as opposed to being absolutely required. So, and in this case, it is absolutely required, partly because it's pulling the strands apart. You end up with a much elongated structure, so that way you can get the 3 strands all interacting with each other and strand exchange to take place. And that also depends on the ATP hydrolysis to have that take place. This order here is actually wrong, and I wanted to mention that as well. So holiday junctions form before you have branch migration that takes place. So what's a holiday junction? That's this crossing over piece here, where this end here, this is your 5' to 3' DNA polymerase, gets hooked up to this 3' end to the 5' end here, and you end up with this crossing over structure, which actually looks much more like a cross, because it's got an internal piece in the middle, and there base pairs form and break. That can move relative to where it first forms, because each time you break 2 pairs, I should say, of hydrogen bonds, another 2 are going to form. And so this branch migration can really move back and forth relative to where it's first formed with no extra energy input whatsoever. And sometimes branch migration can literally go for thousands of nucleotides before you have the resolution of these holiday junctions, which is cutting and putting these pieces back together. You can have 2 different ways that this can happen. One is the crossing over, whereby one of your parental chromosomes is then different at either end, or you can have the patch, where it's just this single little piece in the middle, where you have the different sequence. So here, if we look at this particular case, again, all of the sequences that we have here in the middle, this yellow band, that's all going to be complementary to the red chromosome. And so we've got red chromosome here, red chromosome here. There's a little bit right here, and this bit can be longer or shorter depending on how far that branch migration goes, where you have information from different parental chromosomes. So this is the heteroduplex that I talked about and figured I didn't do a terribly good job the first time around. Yeah?
For the crossing over, does that completely depend on which type of cut you make, horizontal or vertical?
Yeah, so the question here has to do with, when do you get crossing over and when do you not get crossing over? At least as far as the holiday junction is concerned, it's a completely symmetrical structure, because it's really much more like a cross than it is, one piece in the middle and then the 2 continuous on the other sides. We looked at that, we unfolded it last time. And so in theory, that can happen 50% of the time each way. And it depends on not just the one holiday junction. You're always going to have 2 holiday junctions. If both of those holiday junctions are cut the same way, you're not going to have a crossing over. If one's cut in one way and the other's cut in the other way, you will have a crossing over that takes place.
So if they're both cut vertically, you won't have the crossing.
Both cut vertically or both cut horizontally, you're going to have the patch, whereas if one is one and one is the other, then you'll have a crossing over that takes place. So, but at any of these cases, either a patch, which we have here, or a crossing over, there's always going to be a little bit of heteroduplex sequence, which is there. And that's shown in this meiotic chromosome here. We've got all of these potential heteroduplexes which are formed here. One interesting thing that I also wanted to point out here, and this gets back to your question about crossing over versus patches. Strangely enough, and this is not completely understood now, in meiotic crossing over, you only have very few crossovers and many more patches. So in theory, it should be 50-50. But in practice, it's not. And we're still trying to figure out exactly why that is. But it is definitely true that you have a lot less of these complete crossing overs than not. But we're not going to concentrate on those right now. We're going to concentrate on these areas of heteroduplex. So here, again, one of these strands has come from one parent. The other strands come from another parent. They might not have the same sequence. And if they don't have the same sequence, then you can get gene conversion taking place. So, and there's only going to be gene conversion if these guys actually code for different, usually going to be protein sequences here. So you'll have lots of changes that will happen which are silent, again, because of the genetic code. But here, because you now have a mismatch on these two different strands, mismatches are going to cause a problem in terms of your DNA structure. Mismatch repair is going to come in and take one of those strands out and copy it over to the other material. And so what that means is you're not always going to have your regular 2-to-2 segregation process. You might, but you also might not. And that actually does seem to be random, which strand is kept and which strand is not kept. And so that's how you can get gene conversion. It's due to these heteroduplexes which you form between maternal, paternal chromosomes, or for that matter, if you're talking about a haploid, your newly replicated versus your old replicated strand. So that can give you a gene conversion event because of these different origins for homologous. And again, this happens much more frequently if you've got a lot of branch migration that's happened, because branch migration is going to give you more of these heteroduplex sequences. Okay, so that's it for DNA repair. Any more questions before we move on to jumping genes? So jumping genes, transposons, again, why do we care? The reason we care is about 40+% of our genome is made up of these transposons, and particularly the retrotransposons. But I did want to talk a little bit about the non-retrotransposons, because they're really important for antibiotic resistance in bacteria and bacterial diseases, but also because of some of the weird things that happen with some of these transposons. And again, people have asked me some of these questions on D2L. So here, a transposon sequence has inverted repeat sequences at either end of the transposon, these little red sequences. Each of those interacts with the transposase protein, which could be coded in this sequence, but it might actually just be a transposase that's been transcribed and translated from another transposon somewhere else. So to actually get a gene to jump, all you need is the transposase and those inverted repeat sequences. So as long as something's expressing a transposase, then you can have these little pieces move around inside your genome. And that's exactly how the Cre-Lox system works that we talked about briefly in terms of site-specific recombination. So inverted repeat sequences, then the transposase is going to bind to these and cut out the transposon. This transposon is going to go to somewhere else in the genome. It's going to cut at offset sequences, I'll get to in just a sec, in the genome, and then attach this transposon to it. The transposon is a double-stranded piece that has blunt ends, so they're flat. Each end is exactly the same length. However, where you've cut is not exactly the same length. So you end up with these little gaps on either side, and that's what these black boxes are here. These are sequences which are going to be gaps after you've put your transposon into wherever it's going to. That gap then gets repaired by the cellular polymerase. And since these were complementary sequences right here, this is now going to generate a direct repeat right here in the genome where you have the transposon which is inserted. Yeah?
So is the transposase being encoded within the sequence, or?
So the question is, where does the transposase come from? Sorry, I'm paraphrasing here. In most cases, the transposase will be encoded here in the transposon. But that's not always the case. If you have multiple transposons in your genome, which is true like us and most other organisms, some of them will have a transposase gene that's in there, but some of them may have mutated that transposase gene, or it's been deleted, or something else has happened. But as long as those inverted repeat sequences are there, that piece of DNA can be moved. Yeah?
So the transposase is sequence specific, like they look at just, or is it very general inverted repeats?
Okay, so the question is, are transposases sequence specific or not, basically, is another way of looking at it. So the answer is yes and yes. So the specificity is for the binding of these sequences in the transposon, or whatever those inverted repeat sequences are. That's what the transposase is going to bind to. Where it cuts down here, that seems to be rather non-specific. So it's not the same sequence that it was binding to up here. This is the binding to and then cut right next to it. And then once you're in the transpososome, this here is going to find a piece of DNA and cut it and then integrate itself into it. So specific in terms of what it's moving, not specific in terms of where it goes. And it turns out, basically, the same thing happens with retroviruses, only the way you get the viral DNA is a little bit different. But the same kind of thing is happening here. You've got an integrase that binds specifically to either end of your viral DNA and then cuts randomly somewhere else in the genome, inserts there, gives these little gaps, which you can actually see here a little bit better. Those get filled in and going to generate direct repeats. Okay. Then this confusing one. And it's confusing partly because it's not very well described in the textbook. It's also confusing because we don't completely understand all of the extra steps that are going into the replication of these retro elements. I'm going to see if I can get rid of this thing here. Yeah. It's going to confuse me. Let's go back to here. Should give me back my pointer so I can get rid of this eye clicker thing, except it's not doing so. Well, be that as it may. So retro elements, so the non-retroviral retro elements, these are your sequences. Again, almost 40% of our genome is made up of these things. They are DNA sequences which have a poly A tail at one end of them, and of course, poly T on the opposite strand, going from 5' to 3'. These then get transcribed by the regular cellular polymerase. And when we talk more about polymerase regulation after the midterm, turns out that the whole genome is being transcribed all the time. And so it's not surprising that you have RNAs which were going to be now a copy of this retrotransposon which is present in the genome. These retrotransposons, particularly the line, encode reverse transcriptases and endonucleases. These messenger RNAs get processed, they get taken out of the cytoplasm, they get translated, and those RNAs then get re-imported back into the nucleus together with this reverse transcriptase endonuclease. It binds again, just like what goes on with those transposons, the DNA-only ones, the retroviral ones, to a specific sequence in your RNA, which again is a copy of this retrotransposon here. In this case, it's the poly A tail. That poly A tail now is bound. Then this goes and cuts more or less randomly in the genome and gives you a 3' OH. That 3' OH is now the primer for the reverse transcriptase, which is going to make this red copy of this blue RNA that was made again up here by RNA polymerase II. Gets to the end, there's probably another primer that gets attached here by mechanisms that are not completely clear. then makes a second strand, and again, it's because it's like the reverse transcriptases we already talked about, also has a RNase H activity, so it'll chew up the RNA, which is in this RNA-DNA hybrid. You end up with a double-stranded piece of DNA that gets put into the genome. This piece now is going to be the complement of that RNA that was made up here, and then the DNA copy on the opposite strand. And these guys are incredibly common in our genome. Again, over 1 million copies of these things. They're also not dissimilar to the question that you were asking about the specificity. Basically, any RNA that's got a poly A tail that's in the nucleus could actually serve as a template for this. And so you have a lot of the so-called sign elements. We talked about line elements, the long interspersed nuclear elements. Those are the ones that have reverse transcriptase and endonuclease in them. The signs don't have those, but they've still got this poly A sequence that the reverse transcriptase and endonuclease can bind to and copy it into the genome. Okay, so those are the random ones. We also have specific integration that takes place. And this is also getting back to your question about specificity, binding to ends, and binding specifically into the genome. So here, Lambda is just an example of this, but any of the so-called site-specific recombinases, these now are going to bind to specific sequences in what's going to be moved, like the analog we have with the transposon, but they also bind to exactly the same sequence in the genome that they're going into. So here we have repeated sequences. It's a repeated sequence in the case of Lambda, right here, this red sequence, and the same exact sequence in the chromosome that you have integration that takes place in. So this integrase cuts and pastes, but it does it in the very specific sequence that this protein is going to bind to. And so this is the difference with the transposons. The transposons, they're binding to specific sequences in the piece that they're moving, but they're non-specific sequences that they bind to here. Here it's a specific sequence in both cases that then gets bound to. You can also have this case here where you've got sequences, now 2 copies of your sequence in the genome. Those can be bound by one of these site-specific recombinases, and this whole reaction can go backwards. You can cut out whatever's between these 2 copies. And this is what happens if they're in a directly repeated structure. If they're in an inverted repeat structure, turns out when their recombinase binds to and recombines these things, it just flips that intervening sequence around. And if people like, I can draw out the whole process as well, but not now. Okay, any more questions on recombination, site-specific recombination, etc.? Okay, so now, RNA, oh, sorry, yeah.
Is there anything keeping, like, signs, for instance, getting like put in the middle of useful genes?
Sorry, signs? Oh, signs and line elements? Okay, so yeah, the question is basically, what about transposons jumping into random places in the genome? Could they be jumping into places which are a problem? And this is, in fact, a really classic example of the line element has jumped into the gene important for blood clotting and has caused hemophilia. So there are a number of cases of spontaneous hemophilia due to one of these transposons jumping into that particular gene. And so you can actually have these things that happen due to the movement of some of these retrotransposons. It turns out that most of them in our genome are probably not active. They're probably not moving around very much, which is a good thing because we get a whole bunch of them. Yeah?
For the lambda integration, that's just specific to the bacterial chromosome, or is this just using the bacterial chromosome as an example?
Okay, so the question basically here has to do with this site-specific recombination example. In this case, it is specifically Lambda and E. coli. We talked about the Cre-Lox system before, which is also an artificial system. It comes from bacteria, but smart molecular biologists figured out it was a really cool way to modify genes in eukaryotic cells. So they brought in those extra genes and can use it to do genome engineering. Now, there are, as far as I know, no site-specific recombinases in human cells. But again, that's as far as I know, and my ignorance is infinite, knowledge is limited, mine particularly. Yeah?
So the site-specific recombination, that's not part of the three major classes of transposons, right? This is just an extra example?
Yeah, so yeah, the site-specific recombination, this is a much more specific and rarer case that we have. Okay, so no, it's not in that table because they're not that common. But incredibly useful, again, from the genome engineering point of view, which is why we cover them. And of course, they're viruses, so they're totally cool anyway. Which reminds me, yes, you can sign up. If you're a biology major and a senior, you can sign up for classes today. If you're not, you need to wait. Okay, so RNA polymerases, again, switching gears. Now we're starting to talk about gene expression. How do you go from the genetic material, which you just broke, and has been repaired, to now turning that into a protein? First step is to transcribe your DNA, and transcription, basically the same language with a couple of tweaks again, deoxyribose versus ribose, and uracil versus thymidine. But you're basically making a copy, and terminology-wise, the template strand here is the one that you're going to copy. So template strand, it's going to be now the RNA is the same sequence as your non-template strand, with, again, the changes for uracil and thymidine. RNA polymerases are awesome because they don't need primers. They're horrible because they make a bunch more mistakes. And this is my mistake. Speaking of mistakes down here, I had 20 nucleotides per second. It's actually more like 50 nucleotides per second, how these guys go. Which means that they're a lot slower than the DNA polymerases, but still a lot faster than the ribosomes when we get to it. So great advantage, again, no primer required. They can start anywhere as long as you have the right starting place. We'll talk about that in just a second. But they've got lower fidelity and are considerably slower. So how do they get to the right place? Or actually, maybe back up a little bit. This is we've talked about this for replication, for transcription, for translation. You got to start somewhere. Once you've started, you have an elongation process. Then you have a termination step. So you're always going to have initiation, elongation, termination. So for our transcription, your initiation really has to do with finding the right place to start. So that's your promoter. Once you've bound to the promoter, you need to get the two strands apart. Again, just like you have in DNA replication. Helicase activity, pulling those two strands apart. Then, after you've pulled the two strands apart, the RNA polymerase spits out a couple of nucleotides. This is your abortive initiation. And then it decides, hey, this really is a gene that I want to transcribe. Becomes an elongating polymerase. This is now extremely processive till it gets up to the termination site in bacteria. That's a secondary structure which forms in the RNA. And then everything falls apart. You restart and go through the whole process. In bacteria, how do you find the right promoter site? It's through the sigma factor, which is one of the parts of the polymerase. Before it gets started, until it gets into the elongating state, is removed, and you can recycle this whole process. So what is the sigma factor plus polymerase? And so this is also quite different than what happens in eukaryotic systems. It's the sigma factor and the polymerase together that are binding to the promoter. When we talk about the general transcription factors, it's all those general transcription factors that associate with the promoter first, and then the polymerase comes along. So here it's the polymerase plus the sigma factor, also called the holoenzyme. No reason why people decided to do that. So not holodeck, but holoenzyme. Holoenzyme is just the main catalytic parts of your RNA polymerase plus this extra subunit for binding. So binding to promoters, -10, -35. What do I mean by -10 and -35? There's always going to be one particular point at which transcription starts. That's your +1, the +1 nucleotide. And that's going to be +1 relative to that non-template strand. So if you back up along that non-template strand and get 10 nucleotides away from that, it's now double-stranded DNA. That double-stranded DNA, in the case of bacteria, very often has this TATA-like sequence, the -10 sequence. And then if you go another 25 nucleotides upstream from that, so which is, downstream is plus, going away from your transcription start site, upstream is going in the opposite direction. Then you have another set of sequences which is well conserved and also is where you have your holoenzyme binding to. So this is that sequence. See, it's still not working here properly. Not quite sure why. This is one of those sequences here. -35, going from 5' to 3', TTGACA, then, again, 15 to 19 nucleotides. That's from this end of your -35 to this end of the -10. From this end to that end, of course, is more like 25 nucleotides. And then these conserved sequences. This sequence here, TATA, should seem really familiar. Why? Because it's like the TATA box that you see in eukaryotic systems. If it's just TATA, this on one strand, going from 5' to 3' is TATA. On the opposite strand, what is it? TATA. So if you just look at this TATA sequence, and this is true in bacteria and in eukaryotes, you wouldn't know whether this was -10 for a promoter going this way or -10 for a promoter going that way. So it's really important that you have an asymmetric sequence here, which means that it's not the same on both strands. So here the -35 relative to the -10 says your promoter's over on this side. If ever you had a sequence to this side, then your promoter would be over on this side. Those are two different strands. And again, these RNA polymerases can start anywhere. They need to know where to start. How do they know where to start? It's a double-stranded DNA that you have protein binding to. And we'll talk a lot more about protein binding, where all of that happens, again, after the midterm.
So you can determine which direction it's going by where the sequence after the TATA box is? Whichever direction that sequence is after the TATA box is going to be which direction the promoter is going.
So the question here basically has to do with what direction are you going to be transcribing. It's usually not so much the sequence which is what I would call downstream of the TATA box towards the transcriptional start site. It's usually further away from the transcriptional start site. And that's the -35 that you have in bacterial systems. It's the BRE that you have in eukaryotic systems, the TF2B recognition element. Okay, just wanted to briefly talk about the elongating RNA polymerase. Two main things about this. One is that it's extremely processive. So there's been a conformational change, which you can see right here, where there's a part of the RNA polymerase that literally clamps onto one of the strands, and this is the template strand here, that holds it in, serves as a helicase, pulling the two strands apart. You also only have a very short piece here, RNA-DNA hybrid, so where the RNA is attached to the DNA. And that's probably really important for getting termination to take place, because when we talk about termination, it's a secondary structure, sort of basically pulling the RNA. As soon as you pull the RNA out of the polymerase, then you have termination that takes place. So we think about RNA polymerases from all three domains of life. Of course, the archaeal one, most important one here in the middle, is very similar in terms of its structure and number of subunits to the eukaryotic RNA polymerase II. The RNA polymerase 1s and 3s are slightly different than this, but still very much the same kind of structure. The bacterial polymerase overall is pretty similar. It still has this claw-like structure, but it's really quite different relative to these guys. It doesn't have these extra subunits hanging off here on the other side. So eukaryote and archaea, not surprisingly, also use very similar mechanisms for getting to promoters. What are those mechanisms in terms of interacting with promoters in a eukaryotic system? Again, unlike having a holoenzyme that's got a sigma factor together with the polymerase, usually you're going to have association of your general transcription factors and then finally the RNA polymerase. First of those is TF2D because it's got TBP bound to it, then TF2B binding to the B recognition element. That gives you your orientation and says, hey, we're going to be transcribing over here. Then the polymerase binds. We have TF2H with its helicase and kinase activities, which will then allow transcription to take place. And beating your question, Ian, about the other factors which are involved, F, E, and A, are they on here? Right, but have I listed them? No. Does that mean I'm going to ask questions about them on the exam? Yes. Wrong answer. So a couple of things about the Tata box, and one in particular, I think I talked about this last time, which is one of my favorites. Let's see if we can get the lights on here. This is actually not covered, so unfortunately this is not going to be recorded, so everyone who's here gets a bonus. So this was the structure of the Tata binding protein that was originally published on the cover of Nature. What's wrong with this picture? The DNA doesn't look like this. A couple months later, they published the right structure. So don't always believe what you see on the cover of Nature. So switch back over now to here. Just my quick aside. But the main point there is that the Tata box, that the Tata box binding protein binds to, makes this really sharp kink in the DNA. And that process of changing the structure of the DNA is very important in terms of assembling the rest of the complex. And if you remember the table with INR, DPE, etc., those are much less important in terms of transcriptional initiation, but TF2D is interacting with all of those things. Well, how can TF2D be interacting with all of those things? It's because the DNA has been incredibly bent. So that bent structure is actually very, very important for making your pre-initiation complex, getting the RNA polymerase to the right place. Quickly cover all of what Mike Bartlett and his group have been working on for the last 15 years. Transcriptional initiation in archaea. A couple of the important differences here. Many fewer of the general transcription factors, just TBP and TFB. Maybe a TFE homolog here as well. This polymerase doesn't need a TF2H activity, has helicase activity all by itself, and open complexes can be formed and elongated. So a wonderful model for studying eukaryotic-like pared-down transcriptional regulation. Questions on transcription? Usually when he says questions on transcription, he has a clicker question, but I don't have any of those today. Don't worry about it, because we've done those already. OK, so once you have transcription take place in eukaryotic systems, interestingly enough, you don't have all these modifications that happen in archaea. It's also a very interesting question. Then you have modifications of your messenger RNA. First modification that happens literally as the RNA is being transcribed is capping. First thing that happens, which is hidden behind here, is your phosphatase activity takes off the last phosphate. Whenever transcription initiation happens, just like for all of our DNA polymerases, we have a 5' to 3' polymerization reaction. So here, your nucleotide triphosphate, you lose that pyrophosphate, it gets attached here. But there's nothing attached to the 5' end, so you always have 3 phosphates, a triphosphate, which is present at the end of the first made RNA. And this is also true for the bacteria where it doesn't get modified. So first thing that happens is that that last phosphate of your triphosphate, you can see it down here, is removed. Then your, again, my favorite enzyme, guanylyl transferase, takes a guanine and pops it on basically backward onto the end here. So you have a 5' bond that forms here. Then you have a methyltransferase, which puts a methyl group onto this guanosine. So now we have a cap structure. So 7-methylguanosine attached to the cap. In some cases, and in fact, including most humans, you'll also have a methyltransferase that methylates these sugars. Not true in all cases. If you have no methyl groups on the sugars, it's your cap 0, 1 and 2, pretty straightforward in this process. So we've capped our RNA. Once that cap is formed, then the cap binding complex, which is a whole bunch of different proteins, can then bind to that, and that will help get your messenger RNA to being exported, etc. But this has to happen first. If you haven't had this happen first, then the cap binding complex is not going to associate with this. Once it gets out into the cytoplasm to be translated, you're not going to have your initiation factor for E binding to the cap structure. So this is absolutely required for translation and getting outside of the nucleus. Splicing also happens. Almost all eukaryotic genes are spliced. There are a few exceptions to that, but the vast majority are spliced. They're also spliced as transcription is taking place. So you've got a cap over here, your 5' end. Here's an exon. The intron has an A residue in it. That's where your branch point is going to form if you have the association of U2 with it. And the real key here is getting U2AF and U2 attached to this branch point. U1 seems to basically associate with these 5' splice sites as soon as they're made. It's only ever going to splice if there's a U2 which is present here. Yeah?
Can the branch sites, is that variable at all within that sequence of that intron, or is it pretty much in terms of nucleotide length from each exon, is it pretty much the same in each one?
Okay, so the question here basically really has to do with the distance between the branch point and really the 3' splice site. The distance at this end is extremely variable. It can be hugely variable. At this end, it's usually pretty similar, and that's because U2AF actually associates with factors that are bound to the exon, and so it's usually quite close here. Now, that's close in space. It doesn't necessarily have to be that close in sequence. There should also be loops in your RNA as well. But for the most part, it's relatively close to this end. So once you have U2 bound, then you have U4, U5, U6 that we looked at before. These are all of the small nuclear RNAs together with proteins that are bound to themselves first. They'll only bind to one of these introns if you have U1 and U2. Then you have lots of RNA helicase activity. Everything rearranges itself, and U6 has the enzymatic activity which leads to both branch formation and the ligation of these two ends together. Once this is bound together, you now have an exon junction complex that forms here. We didn't talk about this before, but you have an exon junction between these two, and this intron is now chopped up by the exosome inside the nucleus, and now we've got a bunch of ribonucleotides which can be used for the next transcription reaction. A lot of people have asked me about what's all this RNA helicase stuff about. So basically, it's rearranging all the base pairing that's happened between our snRNPs, the intron, and each of the individual snRNPs as well. So one of those is U1 is bound to the 5' end of your introns is what happens, again, immediately as it's being transcribed. Then, in the presence of U6 and these RNA helicases, U1 is taken off of this piece, and U6 will bind to it. And again, these are RNA base pairing interactions that form here. The base, say the branch binding protein, parent needs ATP to get it off, and U2 is being rearranged in order to have base pairing that forms here. You see this base pair here is the same as the base pairing over here. U6, which is base paired over here, is pulling this whole process together. Then you have more ATP hydrolysis, gets U5 into the act, U6, and puts the U6 in exactly the right position to catalyze these base pair, 5' 3' normal ligations to give you your final messenger RNA. Oops. So last thing, and this I've basically covered all of this already, how do you get appropriate splicing to take place? U1 almost always is going to be bound very soon after transcription takes place at the 5' end of some intron. This could splice to this U2, this exon, if U2 is here. If U2 is not here, because you don't have the SR proteins, you don't have U2AF, this U1 is just going to wait until the next one comes along, or wait until the next one comes along, relatively speaking. So U2, and it's really that interaction of U2 together with U2AF, which says this is going to be spliced. How do you get that? Particular exons that are not used most of the time. If you have the SR proteins, just are lots of serines and arginines in them, that exon is going to be used. So now we've done our splicing. Last thing to do is to put on our poly A tail. Poly A tail formation happens again as the RNA is being made. There are specific sequences. No, I don't expect you to remember the sequence, which is here, that CPSF and CSTF bind to, cause RNA cleavage to take place. Neither of these are endonucleases. I went looking before class to figure out what the endonuclease is. It's got a different name in any organism you look at, so that's why we're not talking about it. Once you have this endonucleolytic cleavage, then the poly A polymerase will extend at the 3' end, give you this poly A tail, and the poly A binding protein will associate with this. Once you've had this cleavage that takes place, again, right here, here's the cleavage, there's your poly A tail that happens. Now you have a free 5' end of an RNA, which doesn't have a cap. Caps are protecting the ends of your RNA. This is a non-protected RNA. Now you have a exonuclease. It's a 5' to 3' exonuclease that chews into here and basically, as far as we can tell, catches up with the polymerase. This is also called the torpedo model. So the torpedo is just going faster than the ship that it's chasing after. And so we'll degrade this RNA until you get to this very last little bit. When I talked about the elongating polymerase, it's only very few nucleotides which are holding that messenger RNA to the DNA. And so once it gets to a really short length, that seems to no longer be stable, and it falls off of the polymerase. As soon as there's no RNA which is there, the polymerase is released. It can go back around again. In bacterial transcription termination, same kind of thing. Relatively few DNA, RNA, hydrogen bonds that have formed together. You've got some way of pulling it out, and this way is that secondary structure together. That is stronger than whatever the interactions are with the RNA and the DNA and the RNA polymerase. These come together, the whole thing falls apart. Once you've modified all of your messenger RNAs, then you need to get that messenger RNA out of the nucleus into the cytoplasm so that it can get translated. How this happens, it happens through the binding of proteins. So cap binding complex, HNRNPs, again, there are about 30 of these heterogeneous nuclear RNPs. Some of them bind to introns, some of them bind to exons. So there are actually some which get transported as well. But exon junction complexes, as I mentioned, that tells you splicing has taken place. The main thing that you don't have here in this complex are any of those Ps which you have as part of your snRNPs. So you still have snRNPs associated with your messenger RNA. Clearly there's a problem. So this messenger RNA should not be transported outside of the nucleus. So you can have hnRNPs, you've got exon junction complexes, you've got the poly A binding protein, you've got your cap binding complex. That constellation of proteins will allow you to have export that takes place. Once export's taken place, that cap binding complex gets replaced by EIF4E and EIF4G, and now we're ready for translation. Happy so far? Brains exploding? Yes? Uh-oh. We'll take care of that, don't worry. So just to finish up, last couple of minutes here. Genetic code, again, I like to look at the genetic code this way rather than the way it's presented in our textbook, just because this, I think, gives you a much better idea of what kinds of modifications you can have in your genetic code and still not change the proteins. And not changing the proteins or changing the proteins a very little bit and changing those proteins, the sequence of the proteins, that leads to the structure of your proteins, and the structure gives you... It's getting slow, getting late. So that's the main thing I wanted to talk about in terms of the genetic code. How is the genetic code translated? The genetic code is translated through the tRNAs, the transfer RNAs. These are heavily modified. Again, about 10% of any of these nucleotides is modified in your tRNAs. They're usually made as precursors. They get cut to shorter length. Actual active tRNAs, not surprisingly, because they've all got to fit inside the ribosome. You think about the A site, the P site, and the E site. So they've all got to be just about the same size in terms of being able to fit into the ribosome. They're always in these base pairing structures, and of course they're not clover leafs. They're more like L structures. How do they get the right amino acid relative to the codons, which are down here at the bottom? It's our aminoacyl tRNA synthetase here in light blue, which binds to both the anticodon down here, a number of other bases present in your tRNA, which are going to be specific to that tRNA, and most importantly specific to the amino acid, which is behind the multiple choice thing here. And that's where you get all of the specificity for translation. If the aminoacyl tRNA synthetase puts on the wrong amino acid, the ribosome has no clue. It's going to put that amino acid in any case. So any of the proofreading in terms of putting the wrong amino acid in and taking that amino acid back off, that has to happen at the level of the aminoacyl tRNA synthetase. It doesn't happen at the level of the ribosome. So proofreading, and when I talk about proofreading, it's always going to be going back and taking off that last nucleotide, as we talked about in replication, or here the wrong amino acid which has been put onto your tRNA. Once you have your charged tRNAs, then they've got to get into the ribosome. When we looked at the ribosome structure, when the large and the small subunits are together, it's a very closely packed system, so you've got to have those tRNAs in the middle, and particularly important is getting that first tRNA into the P position in the middle of the ribosome. This can't happen if both of the subunits of the ribosome are together. So in the case of bacteria, it's the Shine-Dalgarno sequence that associates with the small subunit of the ribosome. In terms of the eukaryotic system, the initiator tRNA is present with the small subunit before the messenger RNA actually gets there. But still, you have to have these guys associate with each other before you have the large ribosomal subunit, and then you can plug your way along until you get to a stop codon. The stop codon, there's no tRNA that can associate with that. You'll have the release factors, which will bind to that site, and then have translational termination, so you can go through and do this whole thing again. Got a few more slides on the ribozyme and the ribosome. The ribosome is a ribozyme, but I'm not going to talk about those now. Good luck on Friday.